regulation of transcription. In the prokaryotes, lac operon was used as an example. Why lac operon? And lac operon عنده multiple levels of control and regulation of gene expression. So if you understand the lac operon, it's easy for you, if you encounter issue تاني بالprokaryotes, to comprehend what's happening there. Lac operon, did we talk about lac operon? حكينا انه الدايسكرايد lactose, in order to be broken down to glucose and galactose and imported, يعني there is a group of genes involved بهذا الأوبيرون, والأوبيرون عبارة عن 3 genes under one promoter, and you can only see that بالprokaryotes. فالجين البيتا galactosidase breaks down الدايسكرايد لمونوسكرايد. البيرميز allows the transport of lactose الدايسكرايد into the cell. وترانساسيتيليز كمان plays a role بالmetabolism of lactose. The RNA polymerase transcribes هاي 3 genes in one transcript, in one RNA, which later on gives بالtranslation 3 different proteins. We said a regulatory element not only هو promoter, rather there is the operator. The operator, if this protein repressor binds to the operator, the بيمسك على operator, consequently the transcription is stopped, inhibited. Now if there is lactose, the lactose will take away the repressor and prevent it from inhibiting the transcription. وهذا makes sense. Why does it make sense? لأنه in the presence of lactose, بوجود lactose, I need the 3 proteins to process it. If there is no lactose, what's the point of making the proteins? Right? فin the presence of lactose أو الالولactose اللي بيشتغل عليه beta galactosidase, it will take away, بياخد the repressor from the operator, allowing the polymerase to express those 3 genes وhandle lactose الموجود. If there is no lactose, the repressor will bind the operator. Pay attention إنه the repressor itself is a protein which was transcribed, صار له transcription من gene, I call it lac-I. فالlac Z is translated to beta galactosidase, lac Y gene translated to permease, ولac A transacetylase. Furthermore, we used the operator as an example of cis-acting element. A cis-acting element هو sequence adjacent for a certain gene وinfluences the expression تبع هذا الجين. فهذا cis-acting element. If the gene is far away, it's not a gene هو, as a matter of fact, هو ايش? Element. If that element is far away from the target gene, بصير trans-acting. We talked كمان, we said there is a constitutive mutation. هاي constitutive mutation, a mutation happens to the operator. بصير عندي mutation على operator. Where هو repressor DNA binding protein. والDNA binding protein, not only it binds to DNA, but it recognizes, بتعرف certain sequence of DNA. مش بروح بركب على أي مكان بالDNA. Otherwise it's gonna be a mess. فهذا البروتين بتعرف على DNA وبالتحديد على sequence معين من الDNA اللي هو الoperator. Now if the sequence is mutated, يعني changed, it will no longer be identified, ما راح يتعرف عليه repressor. فtranscription will be always on. لأنه it will not be inhibited لأنه sequence of the operator changed and the repressor can no longer identify the location of the operator. ما بتعرف وين مكان الoperator والrepressor. ليه؟ لأنه صارت في طفرة، والطفرة هاي غيرت السيكونس تبعه، وبناء عليه بطل هذا الريبريسور يتعرف على السيكونس اللي بتعرف عليه بالعادة. فهذا I call it, you wanna call it constitutive expression أو always on expression، نفس المعنى. بوجود اللاكتوز أو بعدم وجود اللاكتوز الexpression is ongoing. repressed أو always off، البروتين itself is، هو البروتين له domains، له regions. إيش يعني domain؟ هي fancy term، مصطلح fancy، معنى region على البروتين. There is a lactose binding region، مش lactose binds to the repressor to prevent it from binding to the operator. If a lactose binding region is altered، إذا تغيرت due to a mutation for the gene that encodes a repressor، the gene اللي بصير منه transcription و translation وبيعمل لي repressor، هذا الجين صار فيه mutation، consequently شكل البروتين بتغير اللي هو repressor، وبطل يتعرف عليه اللاكتوز. So even in the presence of lactose it will not be inactivated. يعني in the presence of lactose it will continue to be active. وactive repressor means what? Means binding to the operator و stops the transcription. Transcription it will be stopped. حد عنده سؤال؟ Any questions؟ أي mutation وين؟ إذا صار mutation وين؟ وين هون؟ هذا الريبريسور؟ مو هاي الفكرة. ممكن يصير في mutation من نوع بحيث انه يبطل يتجاوب لللاكتوز. مش المفروض بوجود اللاكتوز يصير inactive؟ الدي ان اي تبعه المسؤول عنه الجين تبعه صار في mutation عمل لي بروتين. هذا البروتين does not respond to the lactose. ما بيستجيب لوجود اللاكتوز فما بصير inactive بضله active حتى بوجود اللاكتوز. بشكله الطبيعي لازم يكون inactive. يعني your colleague is asking what is the impact of a mutation? If the mutation changes the protein in a way that it no longer responds to lactose, it will keep binding على operator حتى بوجود اللاكتوز and the genes involved with lactose metabolism will be turned off. الexpression تبعهم their expression. بعدين we said there is another level of regulation of expression لthis operon اللي هو cyclic AMP. Cyclic AMP is made from adenosine triphosphate through an enzyme adenyl cyclase. Now it happens إنه adenyl cyclase هذا الانزيم is inhibited بصير له inactivation by glucose. So if there is glucose there will be no cyclic AMP. يعني إذا في glucose ما راح يكون في عندي cyclic AMP. ليش؟ لأنه الانزيم that makes the cyclic AMP is inactive by the glucose. If there is cyclic AMP إذا عندي cyclic AMP the cyclic AMP will activate a protein called CAP which stands for metabolite activating protein. وهذا الactivated CAP will further increase the expression of the RNA polymerase. يعني will increase the activity of the expression machinery. إذا أنا بنشط الكاب هذا بحد ذاته راح يخلي الترانسكريبشن يصير أقوى. متى بصير الكاب شغال؟ إذا عندي cyclic AMP. متى بيكون عندي cyclic AMP؟ إذا ما عندي glucose. And that makes sense. لأنه in the presence of glucose it is better for the cell than اللاكتوز. يعني إذا أنا عندي glucose ليش أروح أغلب حالي أشتغل على اللاكتوز؟ If you look at this image here, in the presence of glucose, the enzyme adenyl cyclase will be inactive and there will be low cyclic AMP. Consequently, CAP will not be activated. ما بصير activation للCAP. والexpression level تبع الأوبيرون بيكون low. In the absence of glucose, I need to utilize the lactose. بدي أحتاج أستعمل lactose. Low glucose allows the adenyl cyclase to be activated. will produce cyclic AMP which in turn will boost the transcription بيزود الترانسكريبشن of three genes that metabolize the lactose وبيستفيدوا منه. Others are the different scenarios. There could be a lactose with no glucose. لأنه there is lactose, the lactose will take away the repressor from the operator, and because there is no glucose there is activated CAP, furthermore enhancing the transcription of the genes involved بالmetabolism تاع اللactose. إذا عندي glucose but no lactose, the absence of lactose means that the repressor will stop the transcription of lac operon, and presence of glucose will keep the CAP deactivated. صح؟ The final scenario both of them are present. وجودهم الاثنين بعطي preferential selection for what? For glucose. Glucose is there. There is low cyclic AMP, CAP is not activated. It's not involved with transcription. There is lactose, a repressor is not attached, is inactivated. Staying away, keeping away from the operator. So there is transcription, but the transcription is low. Why? لأنه الCAP is not active due to lack of cyclic AMP. لا ايش؟ عيد سؤالك مرة. repeat the question. هذا ولا هذا؟ B B عندي both glucose and lactose. Now the preference for the cell is like glucose. So if there is glucose, there is scarce cyclic AMP. Cyclic AMP is minimal, قليل. Therefore the cap is not activated and it's not enhancing the transcription. So at the same time, لأنه there is lactose, it will inactivate the repressor. فpolymerase can function بدون الكاب باعتبار انه الريبريسر is away بوجود اللاكتوز، ولكن هاي الفانكشن is minimal. ليش minimal؟ لأنه الكاب is not active. متى الترانسكريبشن ليفل بيكون higher؟ when there is cyclic AMP in the absence of glucose. منطقي الموضوع. Now regulation of transcription in eukaryotes is more complicated due to the fact that it's not a unicellular organism, especially advanced eukaryotes. يعني بمعنى the same gene under the same conditions I need it to be expressed بالliver but not in the heart, for example. Even though the gene exists in both, لأنه we all came as a multicellular organism from a unicellular zygote إذا بدك. فمكتوب هون الدكتور عمون كاتب لكم إن transcription in eukaryotes is controlled by cis-acting elements, which I hope you know what that means by now. Promoters, البروكسيمال elements والdistal elements زي enhancers. Transcription regulatory proteins زي activators والrepressors that bind to enhancers silencers. يعني بمعنى the same gene in both tissues but the activator is in one tissue but not the other. يعني البروتين. So that gene will be expressed in tissue that carries the activator and it will not be expressed in the tissue that doesn't carry the activator. لأنه الميتوسيس, again I want to emphasize, بدي أركز على نقطة maybe we mentioned. The mitosis is identical daughter cells on the genetic level, but not on the cytoplasmic level. If you split this room into half, the cytokinesis, بوصل الغرفة لغرفتين, what is in this side will be different than what's on that side. في هون مثلا projector, في board, there is trash bin هون, right? فوجود البروتينز in the daughter cells is not identical بالmitosis. And it happens, one of those proteins إنه activators for a certain gene أو repressor for another gene. صح؟ الchromatin remodeling, we're going to talk about it, اللي هو change of chromatin between being condensed and decondensed. DNA modification بصير chemical modifications for DNA زي epigenetics اللي هو methylation of cytosine nucleotides and involvement of non-coding genes. Remember إنه we said there are 22,000 protein coding genes. حكينا في 22,000 gene encode for a protein وفي as much number non-coding genes. وهدول they will discover later on لأنه they carry no proteins. يعني you want to discover them, بدك تكتشفهم through ال RNA تبعهم. Otherwise you cannot detect them. Those genes, even though they do not encode for proteins, هدول 22,000 جين بيعملوا بس RNA, though they control or they regulate the expression, بيأثروا على expression تبع protein coding genes. And we will see that. هذا السلايد, assume this is a coding region, a gene, and there is a regulatory sequence منها enhancers والsilencer in eukaryotes. فالactivators, if they bind to the regulatory element, there is no repressor that binds to the silencer, معناته expression will be high. If less activators bind, the expression will be lower. If the repressor is there, the expression will be hindered, صح؟ لأن the repressor pushes away the RNA polymerase with transcription factors من the promoter region. حد عنده سؤال؟ Let's further elaborate على فكرة epigenetics. Epi, the translation تبعها means above أو فوق. And there are diseases, في أمراض وراثية, known diseases, like Prader-Willi syndrome, Angelman, some types of diabetes, cancer. There is no mutation in the sequence. DNA sequence is normal. Well, the protein structure is normal. What's abnormal هو chemical modifications على DNA منها الميثيليشن. الميثيليشن happens during embryogenesis. خلال مرحلة الامبريو بصير certain pattern of methylation for cytosine مثلا. هذا مش معناه أن كل السيتوسين بصير له methylation. Some of the cytosine بأماكن معينة. Well controlled pattern. If it changes, it might have consequences. وهذا بيأثر epigenetics. It influences the chromatin packaging. كمان it encompasses chemical modifications of histones and DNA. And we will see that. تفضل. بصير مشاكل بالexpression of the genes therefore بتأثر it's not enough يكون عندي normal protein also يكون عندي optimum quantity كمية مناسبة not more not less and at the right time اللي بيسموه spatio-temporal expression يعني بالتيشو معين during certain period of time يعني maybe in the same tissue this gene needs to be expressed the protein had to be present during the fetal development in this tissue but not بالadulthood right هذا temporal different time points spatial expression in different tissues فالidea إنه you need a normal protein not to be defective in structure but that's not enough أخذنا مثال على we took an example about that اللي هو HER2 receptor it's not mutated but it's amplified so there is more protein of it وهذا respond للcell cycle signal بشكل more sensitive صح؟ طيب example that your textbook is talking about هو receptor for steroid hormone now it happens that this receptor has more than subunit or more than one domain شو domain region sometimes each domain has a different function يعني the steroid binds to this domain بركب الستيرويد هون فبسميه ligand binding domain this domain this region of the protein بتعرفوا احنا we all like to use fancy terms لأشياء simple نعمل science and fancy بس domain هو region على البروتين هاي الريجن أو بدي وانا be cool بسميها هاي الدومين binds to the DNA so I call it DNA binding domain this domain the activation domain it binds to other proteins that are involved بالexpression of a gene بيركبوا على بروتينات تانية بيلعبوا دور بالexpression of the gene either increasing or decreasing the expression يعني مكتوب هون what's written here bind to the transcriptional regulatory proteins صح what we just said known as co-activators and co-repressors بروتينات بيلعبوا دور بالrepression or activation of the expression bear in mind خلي بالكم not all of the receptors are present على البلازما membrane تبع الخلية some of the receptors are intracellular especially when the signal can pass through the membrane due to the fact إنها non-polar right إذا السيجنال زي الستيرويد non-polar it has no problem penetrating the cell membrane معناته receptor for it يعني هاي DNA binding domain this receptor تبع الستيرويد means إنه this receptor exists all the way in the nucleus ممكن يوصل للنيوكليوس وبيركب على الدي same receptor attached to the DNA through the DNA binding domain والactivation domain that we talked about it influences بيأثر على expression level لcertain genes in response لوجود الهرمون هذا through either interaction with proteins that influence the transcription or modification of the chromatin structure يعني بالعربي هذا الريسبتور تاع الستيرويد لما يجي الستيرويد بصير له activation فبيروح بركب على الدي والاكتيفيتنج دومين تبعه الاكتيفيشن دومين راح يأثر على يختار جينات معينة يأثر على الاكسبريشن تبعها إما إنه يتفاعل يصير توك بينه وبين البروتينز بيلعبوا دور بالاكسبريشن مباشرة أو بيأثر بطريقة غير مباشرة بغير الكروماتين ستراكشر وإذا غير الكروماتين ستراكشر إذا صار condensed or decondensed means the genes will be not expressed أو expressed respectively حسب إذا condensed بس بطل يصير expression للجينز بالريجن هاي وإذا decondensed أو lose بصير expression